Greetings everyone. Today we are presenting our research project titled Genome Size Evolution and Environmental Adaptation in Eukaryotes. I am Nigi Shikdar and I am joined by Mohammad Zahin Abrar and Sadia Islam. This study explores how genome size differs across eukaryotes and how these differences might be driven by the environments they live in. We combine statistical analysis, machine learning, and evolutionary simulations to better understand the role of genome size in adaptation. Why should we care about genome size? Genome size varies enormously among eukaryotes, from about 2 megabases in some fungi to over 1 lakh megabases in certain plants. But oddly, this variation does not correlate directly with organism complexity, a paradox called C-value enigma. So, what could explain it? One promising explanation is environmental adaptation. Organisms living in harsh or competitive niche might evolve smaller, more efficient genomes. Other in stable or nutrient-rich environments might maintain large repetitive genomes. Understanding these dynamics can shed light on how life evolves in response to ecological pressure. Our research had three main goals. Firstly, to analyze genome size and GC content, which is guanine and cytosine percentage across taxonomic groups like plants, fungi, animals, and protists. Secondly, to use clustering algorithms to group organisms based on genomic features, potentially revealing ecological patterns. Thirdly, to simulate genome evolution under different environmental pressures, testing whether niche specific selection influences genome size. We also built a machine learning model to predict genome size based on GC percentage, coding sequence count, and ecological niche. Let's briefly look at what has already been done in this area. The C-value enigma was introduced by Gregory and others to describe the disconnect between genome size and organism complexity. Plants, for instance, often have large genome due to polyploidy and repetitive elements. Fungi, on the other hand, tend to have compact genomes, possibly because they rely on rapid reproduction and adaptation. Earlier studies used ANOVA to compare genome size across groups and clustering to explore structure in genomic data. More recently, machine learning techniques like random forest have been used for genome size prediction, but not often in the context of environmental niche. Our work builds on these methods and goes further by combining them with evolutionary simulations. For our analysis, we used a publicly available dataset with information on over 500 eukaryotic genomes. The dataset included genome size in megabases, GC content percentage, CDS count, organism group, ecological niches such as terrestrial, aquatic, or parasitic. We cleaned the data using pandas in Python that involved converting all numeric fields properly, mapping organisms to broader groups such as plants, fungi, animals, protists, categorizing niche types like terrestrial, aquatic, parasitic, etc., dropping entries with missing or invalid values. This gave us a robust dataset ready for statistical and machine learning analysis. The rest of the description would be given by my fellow teammate Mohammad Zahin Abrar. Thank you.
Thank you, Nikki. Hello, everyone. I'm Zahim. Now I'm going to talk about statistical analysis of our project. To investigate if genome size and GC percentage differ across organisms group, we performed an ANOVA test followed by Turkish HSD for detailed group comparisons. Here's what we found. Plants had significantly larger genomes than other groups, especially fungi, which had some of the smallest genomes. GC percentage also varied, though less dramatically. Protists had the highest GC percentage, while animals were lower on average. It supports the idea that taxonomic groups and possibly their environments play a role in shaping genome composition. Moving on to the next slide, we can see that we have done clustering in our model. We applied k-means clustering to group organisms based on genome size, GC percentage, and CDS. We used K for 4 clusters, and Silhouse score was around 0.41, indicating a good separation. We also used hierarchical clustering with worse linkage, which produced a dendrogram clearly aligned with known taxonomic groups, as shown in the graph. For example, cluster 1 was mostly plants with large genomes, and cluster 2 had fungi with compact genomes. These results show that genome features naturally cluster in ways that reflect evolutionary and ecological relationships. Moving on to the regressional models, we started with linear regression using GC percentage to predict genome size. But it wasn't very effective. As we can see, R square was only 0.23. So to solve this problem, we used random forest regression model that included GC percentage, CDS count, and ecological niche, rather than just depending on the GC percentage. So in this case, we got R square, which is equivalent to 0.78, and mean squared error, which is equivalent to 145.2. Feature importance revealed that CDS was the most important predictor, maintaining 52% of the data, followed by niche, which is 31% important, and finally GC percentage, which caused 70% of the whole dataset. This shows the number of coding sequence and ecological niche are strong indicators of genome size. I'm going to leave the floor to my fellow team member, Saga Islam, for evolutionary simulation. Thank you.
Thank you, Jahin. To explore whether environmental conditions could directly influence genome size over time, we developed an agent-based evolutionary simulation. The model was run for 100 generations, representing a simplified version of how genome evolution might occur in nature. In our simulation, each organism had a genome of variable size. The genome size wasn't static, it could change from one generation to the next based on mutation and natural selection. Crucially, we linked fitness to ecological niche. This means how well an organism reproduced depended on whether its genome size was well suited or its environment. For example, in terrestrial environments where resources might be limited or where faster replication is advantageous, organisms with smaller genomes were favored. In contrast, aquatic or parasitic niches, which might be more stable or offer relaxed selection pressure, did not penalize large genomes. In some cases, they even favored expansion due to redundancy and regulatory complexity. Each generation went through mutation, modeled at a rate of 0.01, and selection where fitter individuals were more likely to reproduce. So, what did we observe? In terrestrial niches, the average genome size decreased by about 15% across generations. On the other hand, in aquatic parasitic environments, genome size increased by roughly 10%. These trends strongly support our hypothesis. Environmental pressure can drive genome evolution, shaping organisms to better fit their ecological context, even at the genomic level. This simulation allowed us to move beyond static data and into a dynamic evolutionary perspective, highlighting not just what differences exist, but why they might exist. Let's now summarize the core insights of our study. First, we confirmed that genome size varies significantly across major eukaryotic groups. For instance, plants tend to have much larger genomes than fungi, often due to phenomena like polyploidy and accumulation of non-coding DNA. Second, using machine learning, we identified CDS count, which reflects the number of protein coding in genes, and ecological niche as strong predictors of genome size. This reinforces the idea that both biological complexity and environmental context shape genomic architecture. Third, through clustering algorithms, we observed that organisms naturally group based on genome characteristics. These clusters aligned well with known taxonomic classifications such as plants and fungi, suggesting that our selected genomic features hold real biological meaning. Finally, perhaps most importantly, our evolutionary simulation demonstrated that genome size is not static. It can evolve under niche specific selection pressures. Environments do more than just surround organisms. They actively influence how their genomes are structured over time. Together, these findings support the broader conclusion that genome size evolution is not random. It is driven by both ecological and functional forces, limitations, and future work. Of course, as with any study, we must acknowledge several limitations. First, our simulation model was simplified. It used fixed mutation rate and did not incorporate more complex genomic mechanisms such as gene duplication, horizontal gene transfer, or transposable elements, all of which can significantly affect genome size in real-world population. Second, while our dataset was substantial, covering over 500 eukaryotic genomes, it could still be improved. Some ecological needs were underrepresented, and additional genomic matrices like lipid content or non-coding RNA density were not included. Looking ahead, we see exciting opportunities to build on this work. We could integrate gene regulatory data, more advanced mutation models, and even stress-induced genome changes. Another step is to simulate evolution across different phylogenetic branches and timescales to see how genome trends emerge over millennia. Finally, validating these simulations against real-world evolutionary trajectories, for example, using fossil DNA or comparative genomics, should straighten the conclusions and connect our model to biological history. These extensions would make the world more realistic and deepen our understanding of genome evolution as a dynamic, environmentally responsive process. Thank you all very much for your attention. To make our research more accessible and reproducible, we have shared the links of our code and dataset from Kaggle. You can explore the source data, test the models, and even extend the simulations yourself. We are happy to take any questions and or feedbacks you may have. Once again, thank you.